Hi everyone, and welcome to the Stage 1 Biology Cells Test Revision screencast. As we didn't have a lesson on Monday, we are missing out on a bit of time, so what I thought I'd do was I'd put together a whole lot of PowerPoints just to let you know what should be in the test and to give you a bit of an idea on where to start your revision. In case you've forgotten, the test is actually going to be Monday of next week. It is closed book, so it's going to rely solely on your knowledge, and you're going to have 100 minutes. We won't actually be starting the test until the last person is actually in class and seated. Don't forget, it's a summative task, which means that if you're absent on the day, you will require a medical certificate. Otherwise, you can actually be awarded a 0. All the information that I'm going to talk about today is available on Sector, and it's in each of those sub-chapters. I've also put all the answers on Sector, apart from 1.7, and I should have that up there for you by the end of the weekend. Okay, what's inside the test? The test is actually composed of a number of multiple choice questions. There'll be some short answer questions, and there's an extended response question. The best way to work out how long to make your answer is simply by the number of lines on underneath the question. So if you've got 2 or 3 lines under that question, you should actually be making the most of them and getting as much information down as possible. Read the questions very carefully. Make sure that you have an understanding of what explain means and why, and all those kind of verbs. They are very important because they give you the key to how to start your question. Okay, so let's get started. So what I've done is I've gone through, and for each subchapter I've written down the science understandings. These are the things that you actually need to know, and if you don't know these, this is where you need to go back through the text and have a look. I'm actually not going to go through every single one for the 7 subchapters. I'm just going to go through them very quickly so that you have an understanding of exactly what you should know, and hopefully what I say might even prompt your memory. So you need to know that living things are distinguishable from non-living things, and you need to know the cell theory. Remember, there were 3 components to the cell theory. So they're structural and functional units of life. They come from pre-existing cells. They contain hereditary material. And the cell is the smallest independent unit of life, and the cell membrane defines a cell and it separates it from its surroundings. Don't forget, we looked at different types of cells. You might be asked to draw a blood cell or a sperm cell or a palisade cell. So you actually need to know exactly what they are. You also need to be able to know the basic components of the cell. In 1.1, we also had a look at DNA. You may recall that I said that you don't actually need to know how to spell deoxyribonucleic acid. DNA is fine this time. Okay, and then we touched very briefly on the cell membrane. And further down, we actually have a look at the phospholipid bilayer through that fluid mosaic model. 1.2 started looking at the 2 major types of cells. You may remember we have prokaryotes and eukaryotes. You do need to know the difference, and you might be shown a picture of a prokaryote or a eukaryote cell, and you might be asked to actually tell me what type of cell is it. Okay, you might also be asked, what does one type of cell have that the other type of cell doesn't? Okay, in eukaryotes, they're much more complex. So you need to be able to identify all the parts of the cell, you know, stuff like the cell wall, the cell membrane, cytoplasm, ribosomes, nucleus, rough ER, smooth ER, Golgi body, mitochondria, a vacuole, and a chloroplast. Okay, and know the difference between an animal cell and a plant cell. 1.3, we started looking at cell division. So we had a look at what the processes are for mitosis. You might remember the acronym PMAT: prophase, metaphase, anaphase, telophase. You might be asked to actually draw one of those phases, or you might actually be given a diagram and asked to draw the next phase. So make sure that you're familiar with those. We also talked about cytokinesis and diploid cells, and that's where I introduced you to the idea of the chromosomes. And you may remember the difference between chromosome 23. In females, it's 2 X's, and in males, it's an X and a Y. 1.4, cell requirements. That's where we started looking at how cells actually got their energy. You may remember that some organisms can make their own food. Some of them need a combination, and some of them actually have to get them from another source. We also looked at photosynthesis, and there was a word equation. So carbon dioxide plus water will give you glucose and oxygen. You need to know that. You also need to know the word equation for cell respiration. So that's glucose plus oxygen will give you carbon dioxide and water. And the third one is the word equation for fermentation, which is where you've got glucose and that forms into ethanol and carbon dioxide in the right conditions. Okay, we also talked a little bit about lactic acid formation. Okay, that's usually a result of anaerobic exercise, and for that one, glucose is just converted straight into lactic acid. Okay. 1.5, the cell membrane. So this is where we looked at our phospholipid bilayer. You might remember we looked at a hydrophobic end and a hydrophilic end. And you need to understand the importance of the cell membrane and how it separates cellular activity from the outside environment. Okay, and there's a number of ways that things get in and out of the cell, and that is through that membrane. So you may recall this was the start of looking at passive transport, and in other words, diffusion and osmosis. And we also looked at active transport, and I introduced the notions of phagocytosis, pinocytosis. Okay, and the role of the membrane in endocytosis and exocytosis. We also, just to prompt you, this is where we started doing the rhubarb prac. And we also had a quick look at the surface area to volume ratio of cells and how it's actually critical to their survival. 1.6, we got onto the importance of microorganisms. Okay, you may remember I showed you that YouTube clip, and if you go back to the lesson headed 1.6, you've still got the link to that YouTube clip. Okay, and the important thing is that we actually had a look at the types of microorganisms where you have your bacteria, your fungi, your protists, your algae, and your viruses. Okay, and we also had a look at what exponential growth is in bacteria. You may remember that when bacteria increase, they don't actually increase linearly in a straight line. They actually increase exponentially, which means that they double up. So if you start with 4, they both reproduce, you get 8. They reproduce, you get 16, 32, 64, and so on. And that is exponential growth. 1.6 also went through the effects of factors such as temperature, nutrient availability, moisture, pH, and the removal of wastes. So if you've forgotten all that, just head back over that chapter. We also looked at microorganisms as decomposers and also the process of binary fission, and that's where the bacteria basically just split into 2. We also covered in that chapter, and it was a very long detailed chapter, the digestive system and the role bacteria and microorganisms play in that, oxygen production by phytoplankton, and the decomposition of dead and waste material recycles through the... Well, that doesn't make sense. Decomposition of dead and waste materials recycle nutrients through the non-living environment, and recombinant DNA technology. So that's the start of cloning. Okay, and interesting note, humans have actually cultured and used microorganisms for about 10,000 years. So even though they're our enemies when we get sick, they're also our good friends. Finally, 1.7, this is the chapter that you had a look at independently. And it's a very interesting one because it looks at food spoilage and how microorganisms cause food to spoil. Okay, it also goes into looking at hygiene practices and sanitizing, washing our hands to reduce the number of bacteria. We also had a look at ways in which we can prevent microorganism outbreaks with things like how to clean our utensils and our cleaning equipment, and also how to store food. The chapter also goes on to have a look at how to preserve food, and before refrigeration there was a number of techniques that we actually still use today. If you think about your jam, for example strawberry jam, I don't know if you know how jam's actually made. The fruit's cooked and then the equal weight in sugar is added to it, it's heated, it's boiled and then bottled. And that sugar acts as a preserving agent for that jam. So that's one way to actually preserve strawberries. I guess another way would also be to dry them. There is dried fruit, and dried fruit's actually quite tasty, and the drying process removes the water and also stops the growth of microorganisms. Now, I think that is very, very quick. It's only 10 minutes, but that will actually get you well on the way to your revision. We'll be going through this in Thursday's lesson, but I thought that you might want to get a quick little jump ahead and be prepared for next Monday. I'll see you Thursday. Bye.